right, so the story so far that we have in DNA is this. We had a bunch of scientists that first discovered that DNA indeed was the molecule of inheritance. Those were the three main experiments that discovered that was the Griffin experiment. He was the transformation guy. The Avery experiment. He was the enzyme guy. And then there was the Hershey Chase experiment, and they were the virus people. So those three experiments end up proving that DNA is indeed the molecule of inheritance. The next step in discovering DNA is to figure out its structure, because they reason that the structure of DNA is going to have some impact on the three main functions of DNA, of course, which are that DNA must store genetic information, DNA must be able to copy the genetic information, and DNA must be able to transmit that genetic information. So we start to take a look at the structure of DNA. And in the structure of DNA, we have three more famous experiments. Chargaff, he's the base pairing rule guy. We have Rosalind Franklin. She is the photo 51 person. She's the one that actually took a picture of DNA. And then we have Watson and Crick, who actually created this double helix twisted ladder model of DNA. And then that model, they had those hydrogen bonds up the center. So then everybody said, okay, well, then what's the function? Because it always goes back to the three main functions of DNA. The structure of DNA loans itself really, really nicely to the ability of DNA to be able to copy itself. And that's kind of where we're picking up the story. We're going to take a look at DNA replication. All right, so first of all, the structure, like we just said, of DNA allows it to be easily copied or replicated because, again, that is a main function of DNA. DNA has to have the ability to copy itself. Now, each strand of our double helix, because remember, it is a twisted ladder, and just like a regular ladder, it has the two rails on the side and the rungs in the middle. The rungs, of course, are held together by the base pairs, right? So we all know about the A and T, and then the C and G. Those have to base up. Those are the base pairs that go together. Those base pairs are held together by hydrogen bonds. And again, remember, these strands are not identical. They are complementary. I always say DNA is kind of like peanut butter and jelly. If let's say your mom was making peanut butter and jelly for your cousins or your little brothers or sisters or whatever, and then she was like halfway done making all these sandwiches, and then like she got an emergency phone call and she had to hurry up and run out, and she said to you, oh my gosh, finish up these sandwiches and feed the little ones. And then you walked into the kitchen and there was a bunch of bread there, all with peanut butter on it. Well, she was only halfway done. What would you have to do to finish off those sandwiches? What do you have to add? Anybody? You got a bunch of peanut butter. What do you have to add to those sandwiches?
Jelly.
Jelly.
Jelly, absolutely. Peanut butter and jelly are complementary, they go together. Much like the base pairs, we know that A and T are complementary, C and G are complementary. So if you have half a sandwich with peanut butter, you know you have to add jelly. Of course, the reverse is true. If your mom had only put down the peanut butter, or I'm sorry, the jelly, then you would know, oh, I got to add some peanut butter. Okay, complementary. If I have one half, then I automatically know what the other half is. That is what DNA is. The DNA strands are complementary. When we talk about replication, again, and we talked about this earlier in our do now, what we are talking about is the process by which a cell can copy or duplicate its DNA. Remember, we talked about two types of cell division. We talked about mitosis earlier this year, and mitosis is the process of making your regular body cells. like your muscle cells, skin cells, liver cells. If I was a tree, this might be the leaf cells or the bark cells, right? And then there is meiosis or meiosis. And meiosis is the process of making our gametes, our sex cells. So again, in mammals or animals, we say that these might be eggs and sperm. If it was a tree or a plant, we might call this pollen and ovum. But again, these are the cells, the gametes, these are the cells responsible for sexual reproduction. Either way, if I'm going through meiosis or mitosis, I have to replicate my DNA first. And the first step of that is that we have to be able to separate the strands and then use the rules of base pairing to build new strands. All right, so again, we kind of talked about this. Really quick overview. During DNA replications, we're going to take one molecule and we're going to separate it into two strands, right? We're going to unzip it. We are then going to use those strands as a template because they're complementary. So now I know how to build the other strand. And again, those complementary that complementary nature of DNA is going to help me build the new strands according to the base pair rules. All right, quick overview still. So there are four, and there's more than four, but if you want to break it down into the simplest thing possible, we can say that there are four steps in replication. Step one, my parent molecule, and again, step one is right here in the diagram, and it's also available in your notes. Step one of the is when we basically have our parent molecule, okay? We have two complementary strands of DNA, and then each is paired with a specific partner by hydrogen bonding. Okay, and again, it's like peanut butter and jelly. Peanut butter and jelly always go together. Adenine always goes with thymine. Cytosine always goes with guanine. All right, so that's step one. You start with your parent molecule, the one strand, the starting strand, held together by hydrogen bonds with the complementary base pairing. Step two. The next, so really the first step in replication is we separate the two DNA strands. Again, it's like a zipper goes in there and unzips those hydrogen bonds. The hydrogen bonds are broken. Remember, hydrogen bonds are the weakest of the three chemical bonds. Kind of like unzipping a zipper, not really hard to do. And again, now we have two templates. The word template, you might sound familiar to you if you've ever done a presentation. Sometimes you go into your Microsoft Word or your Microsoft PowerPoint or even into Google Slides, and they have these templates where everything's laid out for you, and all you have to do is put in your information. That's what a template is. Well, these are templates because it's all laid out for us, or for the replication process. We just need to put in some of our own information to complete the process. Step three. All right, so we end up with new nucleotides that are being inserted along both sides. Remember, the nucleotide is the monomer of DNA. A nucleotide, the monomer of DNA, is made up of a sugar, a deoxyribose, 5-carbon sugar, the phosphate group, and the nitrogen base. So those three things together make a nucleotide. So remember, we unzipped the nitrogen bases. So now what we're going to do is new nitrogen bases start pairing in. So it's like the peanut butter and jelly. They read it. Oh, okay. This is, and I'm making this up. Let's assume that this is adenine. So I need a thymine. Oh, this is a cytosine. I need a guanine. And again, one at a time, the nucleotides line up and get connected according to the base pair rules. All right. And you can see as this is happening in step three here, the top two have already been, kind of re-zipped up, joined together. These next two are in the process of getting together, and then the bottom three haven't even joined yet. So it's kind of like a zipper. All right, next step, step 4. So now our nucleotides, again, the nucleotide is the building block of DNA. It's made up of a phosphate, a sugar, and a nitrogen base. My nucleotides connect along the phosphate backbones. Remember, we have the backbone, which is made up of the sugar and the phosphate, and these create new strands of DNA. Covalent bonds form on the backbone. Remember, the backbone, kind of like your backbone, is really, really strong. Covalent bonds are pretty darn strong. We build new covalent bonds between the sugars and the phosphates to make the backbone of my new DNA. And then we have hydrogen bonds form between the complementary base pairs. Okay, so we have hydrogen bonds going down the middle, holding the two strands together. Remember, those are kind of weak. And then we have covalent bonds holding the backbones together. And remember, comparatively speaking, they are much stronger than hydrogen bonds. All right, so where there was once upon a time, there was a double strand of DNA at the beginning of the process, we now have two strands of DNA. The DNA has now been replicated. This is the overall view of replication. Okay, we haven't gotten into any of the you know details really yet. And again, each of these is an exact replica of the parent molecule. So we have two perfect copies of the original DNA. Issues or questions? Oop, went too far. All right, so here's the thing. Replication is huge. It is a huge job. We are copying an enormous amount of genetic information. And the coolest thing is our bodies do this with very, very few errors. Usually we have only one error in the base pairing for every 10 billion nucleotides. That's a lot. The replication process is really, really fast and really, really accurate. So in the time that you are going to be here in class today, which is approximately 1 hour, your body, on average, is going to make about 100,000-ish new cells. What that means is, over the course of this hour that you are here in class, you are going to be making hundreds of billions of copies of your DNA. Okay? Because remember, each one of your cells has 23 chromosomes. Technically 46 because, we have 23 pairs of chromosomes. Each chromosome is millions and millions and millions of nucleotides long. And you are going to copy about 100,000 of those in the next hour, which is kind of crazy. And you're not going to make that many mistakes. I think we make more mistakes as humans, like when we're copying our friends' homework, which is kind of crazy. All right. When we have large eukaryotic chromosomes, and again, I want to remind you guys, one moment, guys, I have a visitor. All right, hi guys, sorry about that. All right, so again, in a large eukaryotic chromosome, and again, we have eukaryotic cells. These are the ones that have nuclei. They have a lot more complicated DNA versus the prokaryotic cells, which don't have nucleus, like bacteria and stuff. DNA replications happens at hundreds of points along the molecule. This speeds it up because, again, if I was looking at just starting on one side and going to the other, it would take entirely too long. All right. I'm trying to figure out where we are. There we go. All right, so hundreds and hundreds of points along the molecule. Replication then starts in both directions until the entire DNA has been replicated. So again, we are going to start in hundreds of different points, and we're just going to proceed until it's done. This is how DNA. All right, anyway, sorry, there was an announcement. Anyway, so it's going to proceed in both directions until the entire DNA molecule has been replicated. This allows for a very, very, very long DNA molecule to get replicated really, really quickly. The site where the DNA molecule separates, and I think somebody had mentioned this in their do now today, where the DNA molecule separates and starts to unwind is what we call the replication fork. And there are dozens of enzymes and proteins that are used to participate in DNA replication because, again, this is one of the core functions of DNA, and so it has to be done correctly. Where are we? Okay, cool. We are going to stop pretty soon. All right, so you have this picture in your notes, ladies and gentlemen. Replication begins at sites called origins of replication. Those are just special spots along your DNA where your DNA knows that's where we start. So these black dots in the picture, we can think of those as being replication origins. What ends up happening at those spots is your DNA, again, two-sided. We have these little bubbles that form, and those are where we have a specific sequence of nucleotides. And as they open up, you can see the blue, that's where they are starting to replicate. So that point there where the red arrow is, that is one replication fork. Over here we have another replication fork, etc., and so forth. And you can see these bubbles getting bigger and bigger as more and more DNA is being replicated. Proteins that initiate DNA replication recognize the sequence and attach the DNA separated into strands, opening a replication bubble. So again, that is your replication bubble. And again, because we have more than one origin of replication spot, we have more than one replication bubble. Each replication bubble has two replication forks. Oops, didn't mean to do that. So again, the replication fork is here, 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 here. Each replication bubble has two replication forks. Again, the purpose that we're doing here, we are unwinding and we are getting that the new nucleotides to form. The blue in my picture is representing the new DNA. So the replication then proceeds in both directions until the entire molecule can be copied. DNA in a eukaryotic cell might have hundreds of replication organs. And again, this speeds up the entire process for copying really, really long DNA molecules. So again, we start off with our normal DNA. The DNA has origins of replications where it starts to separate into a replication bubble. We get our replication forks on either side of the bubble, and that is where we start to unwind and build our new DNA. So we can now see in this fourth part of the picture, again, for my picture, it's blue. You have brand new DNA strand, a brand new blue DNA strand matched up with the old black strand. Again, so now we have two brand new strands of DNA. Again, right there, that is my replication fork. And I don't remember if I put this picture in for you or not, or something similar to it. But the replication fork, again, is just this Y-shaped region at the end of each replication bubble where the new strands of DNA are elongating. So again, and then these blobs that you're seeing, the blue and the pink, those are different enzymes, which we'll get into in a moment. But for right now, we are going to pause and we are going to have a worksheet on this. And we are going to do the remaining of the assignment, or the notes, I should say, tomorrow. So at this point, I'm going to go ahead and stop sharing my screen. Give me 1 second to get out of it over here. Okay. And 1 moment, and I will get you the worksheet that goes with today's notes. So nobody really had any questions. I don't know if I was going too fast, but nobody said anything. I had a couple messages in the chat, but they were mostly about attendance, which is fine. I appreciate that. Give me 1 moment. I can get your worksheet.